Protein receptors are defined by their location within the cell. We'll first talk about protein receptors that are inside the cell, the intracellular receptors that are not bound to the cell's membrane. The three other types of receptors are bound to the cell's membrane. Channel-linked receptors, enzymatic receptors, and G-protein coupled receptors all have domains embedded in the plasma membrane. Intracellular receptor proteins do not have a signal binding site outside the cell membrane. That means that the ligand must travel through the phospholipid bilayer to reach the receptor protein. Therefore, the ligands of intracellular receptors are small hydrophobic molecules. When the ligand binds to the intracellular protein receptor, they will turn specific genes on or off. Intracellular receptor proteins and their ligands are important modulators of gene expression. An example of this is vitamin D receptors. The ligand in this case is a modified version of vitamin D, which is fat-soluble. The modified vitamin D travels through the plasma membrane and binds with the vitamin D receptor, or VDR. The VDR then pairs with another protein called retinoid X receptor, or RXR. Together, the VDR and the RXR modify the expression of genes involved in the mineralization of bones. Malfunctioning VDR proteins are associated with the development of osteoporosis. Channel-linked receptors are gated ion channels. This class of receptor proteins are embedded in the plasma membrane. The protein's domain on the exterior of the cell has the ligand binding sites. When the hydrophilic ligands bind to the receptor, a channel will open, allowing ions to flow into or out of the cell. A great example of this is the gamma-aminobutyric acid A receptor protein. This is a channel-linked receptor, which remember are gated ion channels. It's composed of 5 polypeptide chains. When the GABA-A binds with the neurotransmitter GABA, chloride ions diffuse into the cell. This diffuses the difference in the electrical charge inside and outside of the nerve cells, slowing down their nerve activity. GABA-A is an important regulator of sleep. Enzymatic receptors are also membrane-bound receptor proteins. They have just a single pass through the plasma membrane. The hydrophilic ligands will bind to the receptor on the outside of the cell, activating the protein to catalyze a chemical reaction. An example of this is the insulin receptor protein. Inside of this cell is an organelle called a vesicle with a bunch of GLUT4 glucose transport proteins embedded in the plasma membrane of the organelle. When insulin binds to the insulin receptor protein embedded in the plasma membrane of the cell, protein kinase is activated and phosphorylates a bunch of other molecules in the cell. These molecules then move the vesicle with the GLUT4 glucose transporter proteins to the cellular membrane. The membrane of the vesicle fuses with the cell's plasma membrane, and GLUT4 allows glucose to enter the cell. G-protein coupled receptors pass through the cell's membrane 7 times. The ligand receptor domain is on the exterior of the cell. The G-protein, its full name is guanine nucleotide binding protein, which is why we call it the G-protein, interacts with the receptor protein on the interior of the cell. The G-protein has 3 subunits, alpha, beta, and gamma. When the ligand binds to the receptor protein, the GDP on the G-protein is phosphorylated. The alpha subunit of the G-protein travels to another enzyme. There, the alpha subunit is dephosphorylated. Secondary messengers are then triggered at this stage.